to test the adsorbed amount of cellulose in the initial stage. And we can find in a lot of papers they test the adsorption amount using a sampling way. Sampling is like this. First, in a slurry of the biomass, they will add the enzyme. Then use a method to withdraw the hydrolysis liquid at a given time. For example, in the first 1 minute, first 2 minutes, or in the first 3 minutes, we will get a sample. After we get the sample, we test the change of the protein concentration in the liquid, so that we can get to know, okay, in the first stage, in the first 10 minutes, how many cellulose have been adsorbed by the substrate. The adsorption ability highly determines the final hydrolysis conversion rate. To study the adsorption of cellulose, we have another reason. This reason is that sometimes cellulose not only adsorbs on cellulose substrate, but also adsorbs on lignin. Lignin is also a solid state particle. It can bind with cellulose non-specifically. So if we study the adsorption capacity, we can get to know that, okay, which one has the higher affinity to our enzyme. It is important for us to characterize the affinity, different affinity to the inhibitor, to the substrate. To study the initial adsorption, we have many methods, well developed. The first one is very simple. It's sampling. Okay, at the given time. Okay, maybe 1 minute, maybe 2 minutes. Then we test the change, the loss of protein in the liquid. We can use a nitrogen analysis. It's very simple. And we can also use a LORI, Bradford UV. All of them, they work. However, we can only get a few data at a given time curve. We cannot get time resolving. Time resolving means, okay, in the first second, enzyme adsorbs on substrate. In the next second, we get the data. Okay, it has a very tiny time curve. So I'm trying to develop the time resolving method. The time resolving method, we also have a method where we developed. This depends on the microbalance. Microbalance, it is a quantum crystal microbalance. This microbalance is very sensitive to the change of a membrane, of the cellulose film. On the cellulose film, if a cellulose deposits on the surface, it can record the change of the weight. However, this equipment is very expensive, yes, and it only applicable for the cellulose film, and cannot be used for the fiber, because fiber is not smooth, does not have a smooth surface and the thickness, and it is difficult to control. Okay. So that's why I'm going to do this study. It is a challenge. In technology, it is a big challenge. Okay. I start to use spectroscopy method. Spectroscopy method is a time resolved, because in UV system we have a detector. It records the data very fast. Okay, in a few seconds, in 1 or 2 seconds, and it is used for the dynamic monitoring in chemical or biochemical processes. For example, we test the activity of lattice, activity of other. We can use the time resolved spectroscopy. Okay. In this system, for the cellulose adsorption, we have at least 3 technical challenges. The first one is the lignocellulosic fibers. It does not dissolve in water. It is a long fiber. It may cause very heavy interference to the light, absorption. Okay. It causes light absorption and it causes light scattering. Another problem is the fine fiber. Fine fiber also causes strong light scattering. And the lignin. Lignin in biomass, if you put a lignin in biomass with lignin in the water, it always leaches the new lignin into the water. The lignin can migrate from the solid to liquid. Although the amount is a very small amount, but it causes interference with light detection. So we have three technical challenges. And I provided some proof. This data proves that, okay, this is pure lignin, pure protein from novozyme with EG, glucanase with CBN, yes. It has a band, a specific band, and a peak at around 280 nanometer. 280 nanometer. But for biomass and cellulose, this is cellulose. Cellulose generally it's pure. It's from field paper. It's pure. So it causes strong light scattering. From in a wide range, it causes light signals. Okay. And for other biomass, they causes not only the light scattering, but also the light absorption in the UV rating. UV rating because of the leaching of lignin. Okay. And if we mix the cellulase and the cellulose and the biomass together, we will find that at a different time the signals change. Okay. Using an UV visible microscopy, we can get the spectrum at every second. Every second it produces one spectrum. Okay, I developed apparatus like this. Yeah, I like to develop a new apparatus. Yeah. It is here in this flask. Okay, and here is a magnetic stud. Cellulose or lignocellulose biomass, the deposit in the flask with A9, with cellulase, with pure cellulase, and it's blend, it stirs. Okay, then I use a pump. To withdraw the containers, withdraw the liquid from the flask, and I use a screen. Use a screen like this. Here, this screen is made of steel. Okay, it is strong, not easily broken, and it has a very tiny hole in the screen. Okay, and it prevents the lignocellulose fibers. All of the fibers will be prevented by the screen. Okay, only very tiny fiber, fine fiber, can pass through the screen and flow through the pump, flows in the tubes. Then in the commit cell, all of them, including the lignin, including the fine fiber, and including the cellulase, they enter the cell and they detected by a UV photospectrometer. Yeah. So the screen is very helpful to prevent the interference of a large fiber. Yeah. Then what I'm going to do is to try to calculate from the spectrum to see which signal belongs to protein, belongs to the change of cellulase. And then I developed a dual wavelength method. Dual wavelength method is very simple. We test two wavelengths. The absorption of light at two wavelengths, λ1 and λ2. And we assume that the total absorption of light is caused by absorption of protein and absorption of this is interference factor. Yes, we combine the fine fiber and lignin together. Yeah, we name it as interference factor. Then we use two coefficients. to express the absorbance, and we use Lambert-Beer law. Lambert-Beer law is also very simple. Finally, we get an equation like this. Okay. After the mathematical transformation, what we should do is to determine all of these coefficients. These coefficients is very easy to get. For example, this one. This one is the epsilon of coefficient of protein. We can test it use a pure inline. And this one tau and zeta and tau. We can test a pure one. Use for example, if we use cellulose, we test the cellulose without any inline. We can get a spectra, and we can get a lot of spectra like this. We can get a lot of spectra like this, and we calculate the slope. Calculate slope, we can get the coefficient. So all of the coefficients we can get. Okay? We can predetermine them. So finally, what we are going to do is to calculate the change of content of protein of the inline from different wavelengths of absorption. Okay. So these are the coefficients we predetermine in my lab. These are the change of absorption at different time curve. Every 7 seconds we get a signal. Okay. And use these equations, we can calculate it like this. These plots are very good, very smooth. Okay, very smooth. From the 0 point to 60 minutes, we can get very fast steady state at about 6 minutes. Okay, so the absorption is very fast, very efficient. And we also compare the cellulose absorption on cellulose and the absorption on aspirin. Aspirin has a much higher absorption ability. Yeah. Aspirin is biomass. Yes, pretreated. Yeah, pretreated. And next, I asked my wife. My wife works on mathematics to help me to fit all the plots using an equation, using a well-developed kinetical model. And we found that we tried a lot of different models. This one fits very well. So finally, I get a conclusion that the endoglucanase is more rapidly absorbed on pretreated aspirin than the filled paper fiber. We have a lot of following papers, but I am not the co-author because I left the lab for at last. So I did not. The following work I did is about another enzyme. This enzyme is also an endoglucanase, but it does not have a CDN. It has only catalytic domain. Yes. So its binding ability is poor. Not as good as EG with CDN. I got absorption curve like this, not smoothie. As it absorbs on the substrate for 5 minutes, desorption, sudden desorption occurred. Then, in the following 5 minutes, desorption. I tried my best to explain it. I used micro AFM atomic force microscopy to observe them, observe their binding on the surface of cellulose microfibrils, and proposed that an EG without a CBM, they form chlorides, they form associates, aggregates of inline particles, and as they absorb on the surface, they cannot move along with the substrate. But the CBM can direct the specific binding and the progressive moving. But those are without Those glucanases without CBM, they tend to form a colloidal particle, a large particle, and the desorption is then occurred. It was in this year, in last December, we developed another research calculation method. This method is originated from a very old question. This question I asked to a lot of students. Okay, if I have a glucose aqueous solution, glucose water solution, the glucose, the mass of glucose is 10 grams, and the total volume of solution is 1,000 milliliter. That's the content of glucose. Okay, it's very easy. We can easily get it 10 grams per liter. But if we have a biomass slurry mixed with glucose, and the glucose in the supplement 10 grams per liter, and the slurry total volume is 1,000 milliliter, can we get the glucose total mass? We tried to solve this problem, and we found that the glucose content, the mass of glucose is not 10 grams. And what's the accurate glucose mass? We are trying to solve this problem. First, we use the method provided by NREL. NREL said, okay, you do the analytical hydrolysis. Okay, you take the biomass into consideration because it occupies the volume. It has a density very close to water. So if you are going to do that, if you have a glucose content in the supplement, and if you have a total volume, you can do that. But do remember, the volume including not only water but also the biomass. So it is better for us to do the test of gravity. So you use NREL method. And another method, if we are going to use, we have to remove the biomass from the total volume. Okay, we remove biomass, leaving only liquid. Then we can test the properties of the liquid. Liquid is glucose solution. We can test the parameters there. So I compared them. I compared the NREL method and the liquid volume calibration method, and I found that if I use the NREL method, this blue line is from the real glucose mass I added into the system, and I find that there is an overestimation if I use the NREL procedure, and a less estimation if I use the liquid volume calibration method. So I developed a new method, and I concluded that glucose distributed in water and biomass. Both water and biomass, and uniformly. We can take the biomass as, for example, activated carbon. It also absorbs the glucose, but in which part glucose has a higher concentration? For example, glucose is enriched in water or enriched in biomass. We don't know. We don't know. Yeah. So my research starts from this question. I can study the well-established method from Christensen. Christensen published a paper almost 10 years to 9 years ago. He do this research like this. First, he do the separation. Separated the hydrolysis. Separated hydrolysis. Solidly, he wash it. Thoroughly wash the solid and dry and weigh the solid mass. So finally, he can get a real volume of the total mass of the liquid, okay? Because he has known the solid residuals. Then he tested the sugar content and the gravity in the liquid. So finally, he can get a very precise data using this method. However, I asked my students to do the study, to do the measurement, exactly follow this procedure. and my students complain a lot because it causes a long time. Usually for 1 day, we can get only maybe two data because of washing, because of the washing. It requires several hours to do the washing, to do the drying, and to do the weighing. It's very good. It's a very precise data, but it provides precise data, but time-consuming and laborious. Then I developed a multiple dilution method. Multiple dilution is I have a slurry of from the analytical hydrolysis of biomass. Then I sampling 1 gram slurry to this beaker, and I add water in. The water for each time I add the same volume water into the slurry. Then I can get the supplement. I can get different content of glucose. All of them. I can test them. Okay, not difficult. Then I developed a lot of calculation method from this. First, after dilution, okay, the total mass of glucose does not change. Okay, but the concentration of glucose changed and the volume changed. Then after the mathematical transformation, I get this. And if I do multiple dilutions, a lot of dilutions, I can get this. Yes. So what I need to do is to test the different volume and different concentration. I do not test the gravity. I do not need to wash. Yeah, I do not need to dry to weigh. It saves a lot of time. But in order to thoroughly, to extensively study this problem, I do assumption. This assumption is very important. I assume that all the liquid in the slurry can be divided into 2 parts. One part is free. It has a free volume, and another part is absorbed. I assume that glucose cannot be absorbed by biomass, but the glucose concentration solution, only glucose solution can be absorbed by the biomass. Now I get I use a coefficient K to express, and I can derive a new function, a new equation, okay. A new equation like this. What I need to do is still I need to test the change of the concentration of the slurry. Okay, it's not difficult. And from this I can derive get to know the total mass of the glucose. It is a reciprocal correlation method. The right figure presents the final data from this method. Okay. And I test a lot of different slurries with different amount of glucose added. Okay. It fits very well. Then I tested a glucose yield from a different solid loading, high solid analytical hydrolysis of biomass. If I use the envelope procedure, the ethanol yield is very high, very good. But if I use the liquid volume calculation method, the data is very poor. It's less estimated. And the Christian's method is okay. And this is my multiple dilution method. Fits very well. Since in the SSF process, there is not only glucose but also ethanol. Ethanol will change a lot. It changes the gravity. Ethanol changes the gravity of the total of the components. So we have to discuss the interference of biomass interfered by the ethanol, the content of generation of ethanol. So I tested ethanol content in the slurry and see that it does not influence my method. No big influence. This data were from the fed batch process of pre-treated dilute acid pre-treated corn stover. Yeah, I use the method, multiple dilution method to calibrate, and finally I get to know that okay, after the third batch I can get glucose concentration, a very high glucose concentration, but the glucose mass is here. I get to know. I also tested the K. The K determines the distribution of glucose in water or in biomass. I found that glucose distributed more in water than in biomass. This method can also be used to test the distribution of ethanol, distribution of xylose, distribution of butanol. I compared them. The result is that for butanol is enriched in biomass, less distributed in water. The line for butanol is higher than 1. If they distributed equally, uniformly, it should be here. So this method is helpful for us for discussion. Yes. Thank you for your attention. Thank you for your listening. So do you have any question about these two methods?
This method is applicable for both volatile and non-volatile, both volatile and non-volatile, both types of solvent, water and ethanol. Ethanol is volatile.
Yeah, for water is okay. For ethanol is also okay. For the fermentation process is also okay.
Binding to biomass is same with water as
ethanol there is slightly different. But ethanol is just lower gravity. That is lower. So we have to discuss the interference by ethanol. Ethanol content is slightly, but use my method it does not increase. Because this equation, I test only the change of glucose concentration. I do not test the change of volume. Yeah, because for each time dilution, I use the same volume of water, the same. Like this method?
Yeah. How can we apply it practically in the biomass production? Practically. How is it going to help the biomass production?
Yes. If we are going to do the analytical hydrolysis at a very high solid loading, For example, 20% to 30%. Okay, this method is very good. But if we are going to study at a very low, for example, 2% or 5%, unreal method is very okay. So it is particularly developed for the high solid consistency conditions. Yes.
So it is, you have used this method.
Yes.
And you have compared with other methods.
Yeah, with other methods. Yes.
Okay. So you published recently.
Yeah, I published it.
Oh, very good. Congratulations.
It has been accepted by Analytical journal.
Analytical.
Yes.
I think you have finished your tutorial.
Yes, I finished.
Okay, no problem. So meanwhile, we can discuss and start the further procedures. So you can say, and I think you can inform them about the evaluation first. No, we'll discuss. We keep discussing in the process. Participants, they can start their group discussion and whatever procedure he is telling, Dr. Keshav. Meanwhile, I will discuss with Dr. Liu. Okay. There is one important announcement for faculty members present here because we after 4:30 we have to go to the department. regarding one important program of the university, that is the student election. So we have to enter inside the department before 5. Otherwise, 5 they will close the grade. Okay, so we have to be sure that we finish this session by 4:30. Okay. So I will keep discussing. You start the procedure of discussion and all those things. Dr. Liu, I was just asking you about just the previous slide.
Yes.
This one. You have in your team some mathematical person.
Yes, yes. My wife helps.
Oh, your wife?
I cannot do mathematical calculation like this.
Your wife is there?
Yeah, she helps me a lot.
Okay.
See, use this to build up this kinetic model.
Okay.
I cannot do the calculation, and she help me.
Okay. And
we tried a lot of different models to fit the data.
Okay.
Yeah, to fit the data.
And whatever things we are doing, like cellulose as pen.
Yes, cellulose as well. Different.
Yeah. Different.
Different, yes. I don't know why, but actually, the pre-treated aspen has a higher absorption capacity to cellulose.
Okay. Please have seat. We will have the discussion because there is, the participants, they are also discussing on various aspects. And the calculation part, we need to depute some mathematician. Like in your team, your wife is there. Okay, she helps you. But I think for our students, we need to contact some mathematical person or a chemical engineer can do this linear expression and all that.
Yes.
The chemical engineer can do that.
Yes.
He can do that. Okay. And this, I think, yeah, no, I don't know. For me, it is very difficult.
Because everything software or Excel works.
They can go. Okay. How? All these, whenever you are using these dual wavelength method, there is also where you publish this one.
Many other ways.
Many others.
There are a lot of applications.
Okay. Can you go back? I want to see some slides where I have some queries. Oh, this technology developed by you.
Yes. Yes.
Okay.
I invented it.
Oh, very
no
good.
patent.
No patent.
Yeah, in the US.
Okay.
It depends on the use of screen, metal screen. Yeah.
Okay.
To filter, to stop, prevent the penetration of lignocellulose fiber.
Okay. Okay.
So that only lignin, only very tiny fiber and the protein can enter the tube and can be detected. What I'm going to do is to use the calculation method.
Okay.
Use this calculation method. Okay. To reduce the interference of lignin, interference of fibers, and you get the real data from protein.
Okay.
The concentration changes,
Okay.
concentration decreases due to the absorption. So I can test it every second, in every 7 seconds.
Oh, you show this. Very interesting. This one.
Yeah, every second, 7 seconds.
No, this.
A lot of data.
We can enlarge this one.
Yes.
And we can dance and see what is this aspen, cellulose aspen.
Yes. They have some kinetic data. Kinetic data is constant, absorption constant. We can compare that. Then we can compare the affinity.
Okay. Okay.
Affinity is allowed. Yeah.
Okay. Okay.
We can also connect them. See this inline structure. For example, this kinetic without CBM.
Okay, without CBM. And with CBM also you have.
Yeah, the CBM, this is
CBM.
very stable, very smooth. But this one,
Without
absorption
CBM.
and dissorption, absorption and dissorption.
Okay. So once you modify and keep the CBM, it works well.
We choose the enzymes from Novozymes.
Novozymes.
Most of them from Novozymes.
So Novozymes, they have some establishment in China also.
Yes, a lot.
A lot.
In Tianjin.
Tianjin.
Tianjin, they have factories there. enzyme factory.
And they have people from China and India and all places. Only Chinese people.
A lot of Chinese engineers.
Oh, a lot of Chinese.
Work for a long time.
Okay. And I think this is very interesting. You just go back previous slide. Yeah, this one. EG with
the sorption of EG with CBN.
CBN. Okay.
On cellulose.
You can have seat, please, because I'm just seeing the pictures.
Besides the CBN, the EG is presents as an individual particles. It presents with no CBN.
Okay.
Colloid.
Okay.
A lot of particles together.
Okay, means you will see the colloid from here without CBN, is it?
Yeah, without CBN.
Okay, and that is with CBN. So this distance your software or something?
Yeah. This is cellulose micro. This is inland particle.
Okay. And this also you have already published? This one?
Yeah.
Okay, which journal you have published?
EMT.
Enzyme and Microbial Technology.
Technology.
Very good journal. EMT.
All the comments are very positive.
Okay.
Very positive.
And you did a very good work, so comment will be positive. Again, you just go to scroll through some more. Yes, keep it here. Direct the specific binding on cellulose, prevent the form of colloidal particles for age. The progressive moving of cellulose rather than fast deabsorption. Okay. Next, please. This glucose biomass.
Yes.
Okay. Can you explain this equation?
Yeah, this equation is very simple.
What does it mean? I don't.
If I have a glucose in water,
Okay.
this is the glucose mass, glucose weight. This is the solution volume.
Okay.
I can get to know the content, the concentration. Very
Okay.
simple.
So you are getting mg.
Yeah.
CF?
Mg is the mass of glucose.
Okay.
CF is the content of the free glucose in water, and this is the volume. It's very
Okay.
simple. But if there is a biomass,
Okay.
totally different.
Okay.
Totally
So
different.
you can have the, like we discussed the tutorials about the metabolic engineering and other aspects which I was covered. You had some questions you wanted to ask.
A lot of questions.
So you can ask few of them because we have 15-20 minutes time. Yeah, you can do that. You have your notebook?
Yeah, I have looked.
So how? If you have anything which we can discuss right now regarding the sections which you were asking.
First, I want to try to screen inhibitors for cellulase.
Inhibitors of cellulase.
Because when we're going to use cellulase
Okay.
in pulp milk, it is very difficult for us to stop the action of cellulase after banana. Generally,
Rajesh,
we cannot use heat. We
Yeah.
cannot use
Can you go there? Yes, please. Just you can sit also here. You just sit.
For
Cellulase inhibition.
for cellulosis.
Yes.
We actually normally do we characterize the design in terms of its inhibitor also.
Okay.
And in terms of organic solvents levels in already. So it's a really work that we do regularly in our labs. We have done for lactases. We've done for proteases. But for cell bases, what I would suggest that if if you're interested in getting this done, so we would procure what kind of cellulose we're talking about. Pure cellulose.
Pure cellulose or the complex.
No, you are taking pure cellulose.
We use cellulose.
Cellulose.
It's a mix.
Okay.
Mixture.
And you know about GIAN? Buckman enzyme.
Buckman products are not pure.
Not pure.
Not pure. Buckman do not produce the original enzyme. They produce from non-line.
Oh.
And then they combine them with Buckman. Yes.
So that is not a pure one.
Yeah.
So is it possible to get a sample of the enzyme that you plan to use in your
Yeah,
so in
simple.
that case we can work out the most strongest inhibitor because we have it in the
Because in the past we cannot use a large amount of chemicals to kill the cellulose. For example, like the metal, like the heavy metal, it's impossible because they are going to produce people from
If I'm just asking a very simple query, if we give some small projects of like calibration of glucose in ice solids to our students,
students.
so is it possible to how much time it will take for complete experiments? To
Maybe for 1 day.
one day they will get it for one product, one enzyme.
Yes.
And this comparison glucose biomass slurry and its structure can also be seen.
Yes.
You can see that.
You can see that. It tells a lot because the glucose has a higher affinity to water and less low affinity to the biomass. But for ethanol, for butanol, it has a higher affinity to biomass rather than to water. They're different, especially butanol. I think butanol.
this study can be done like the finding out the most suitable inhibitor for cellulators, and
Yes.
that is also not toxic to the environment.
Also, yes.
So, yeah, many inhibitors known for enzymes,
Okay.
and like from my parent labs where I was previously, so these were already procured in the form of chemicals. So what we need is a pure sample of cellulose, and we can check the inhibition activity after addition of the right conception.
So
Then we would need the everyday's.
right now.
Yeah. That can be done like as you said, then we will be collaborating in future also. So we can definitely check the.
I also want to do the simulation work about the especially the branch, and the circle.
So this is the structure available with you. The structure.
I have the sequence. I have the sample. I have the sample, but I do not know how to do the simulation.
And also be taken care of by your
How about the
yeah.
I have the sequence.
So how? Okay, you are discussing.
Yeah, we are discussing. Very important question.
Actually, he is also.
Yeah.
Yeah, please discuss.
And we're using this to simulation.
Asking this to take it and we do it for the salts. There are several things that you would like to
Generally, it's copper. In that case, there is we publish it. You put it for two coppers, one fluorus, and one zinc. Okay, not four coppers. Not the four coppers. We get that is
most of the workers have done once and then it's purified when you scan it. So there's a shoulder at 330. 330 that indicates the presence of overlapping like concentration. But by what method did you show that there is iron in the same
resorption, yeah.
Okay. Was the preparation good? Was the preparation good? Then that preparation.
I must have been story the state. Communication.
Preparation was that like
it has been the sequence of the N-terminal, C-terminal, or we cannot get it. The full sequence. We get only small piece of the sequence. You can negative sequence.
this part we should have the pure preparation. They were available with Sigma, but the one you were using, I don't know, you said sometimes it's by Chinese, what you use, we can have that, or we can secure that. So you have to make that from the
तो आप नहीं हम सकते उसको कितनी क्लास हुई ये तो मैं आपको नहीं सिखाऊंगा ना मैं आपसे जितना सिखाऊंगा आपसे Basic classes, so divide them more than 25 some आपको
September. Small troubles. April, might shut up 1 month and sometimes 2 months. Shut on structure.
You can come this side. Actually, this is a discussion going on. You can come here. You also have some queries. Related to that.
I will cover the temperature.
I'm resuming.
Yes. Especially maybe we take so all the notes are in our and the micro doesn't close the properly.
What cutting do you? You just discuss the point.
Yes.
Yeah, this is और वो बच्चों को ज्ञान का बता दीजिएगा फूड वाला ज्ञान का बच्चों का नहीं होता वो बच्चों को बता दीजिएगा पार्टिसिपेट Rajiv, I think I can also listen your discussion.
Yeah.
No. Yeah, it's live.
So I was discussing about the two projects, small projects that we and he collaboratively wanted to do. First is that to find out the individual soft cell bases, and the second was doing the simulation study of protein sequence that he knows. So what I have told is that these experiments are routinely done in the like if you want to look for a probable or the most probable individual for cell basis, so we can find those inhibitors for you.
So, have you are buying inhibitors also? No, you buy it from somebody, or you have isolated. Means from
For cellulose, I do not have any good individual.
Okay, for cellulose. For other enzymes, you have.
For others, lattice I have.
Because
Lattice has a active site. It has a copper. I have some agents.
And lattice inhibitor also you have.
We have tried many things. Cytophenol, we have tried. We have tried beta-lactam, but his requirement is that because he is going to use that paper and pulp industry. So it should
The amount of inhibitor should be very small.
So it shouldn't come as a product itself. If the residues are left in the product, then it's going to be a problem. So with that,
I have few requests from me. Like your lab is working on cellulose,
Lattice,
yeah. So now you have also tried to work on this innovation center.
innovation side.
Yeah. So and then you are working on the different projects related to the cellulose based phase.
Yes, yes.
So is any type of enzymes which you are using, they are isolated by your group?
Okay, okay.
Yeah. Means whether you have the microbes which is giving that particular enzyme, which is producing that enzyme. This is first thing. Okay, first. Yeah. Second question is that if microbe is not belonging to your lab, then from where you have got those strains? Yeah, isolates or So those isolates, how you are maintaining them is you are taking permission or something like that. You can take time, no problem. You can take time because it's a very complicated thing. And in our labs, in Indian labs, we are isolating microbes. So most of our students, they isolate the microbes, and then also they do a lot of screening. Yeah, screening for getting enzymes. And if you visit, I will show you after some time, or rather, Rajesh, we can close a little bit earlier. We go to lab and show those to the participants as well. That will be good. And we are having lot of culture collections. So most of the Indian labs, they do a lot of culture isolations, like such concepts. Can you go back on the previous slides? I have shown the slides. I will just. Okay, coefficients for different lignocellulose structures without each. So this you calculated yourself by the formula. This one, tau.
Yeah, tau.
It's tau.
You test and calculate.
Okay.
Test.
So, and
A
this
lot of them based on the name.
okay. So you have it.
You can see that we have a lot of
Okay.
after the
Yeah, please.
running.
Thank you. Thanks for the question.
This he is telling this technology.
This is useful.
Yes, and he has just gone for some water.
Okay.
So can
The one in the last slide where he said that this is not talented.
this one
No. When he was using the
where in the sorry
okay, he was using the particular membrane. For one.
this one.
No, it was in the previous talk.
Oh, previous laptop. Okay.
So that was really interesting. Like
No, this is also interesting. If this something we don't know about that.
This is a new method that he developed for the quantification of the interaction.
Might be they have published, so we cannot get patent.
Yeah, this is a
See, this is there where you can see this Liu 2010.
Right. Yes.
So maybe if this apparatus is already published, it is not patented. And I think participants they can see and in the lecture notes they can get idea about that. But it's a very small setup. Anybody can do it. We are interested in this. How? Yeah, we want to have such setups in our department.
I'm going to the metal screen.
Yeah, okay, no problem. Means because we have UV based spectrophotometer, and I think pump also we have peristaltic pump. You have?
Yeah.
Yeah, and then we can have some experiment based on that. And one thing also, which might these things I think we have to learn through some. This is the calculation, right? Mathematical calculation.
This we are very weak at, like.
Yeah, because no, his wife. You told that his wife is a mathematician.
Yeah.
So
That's
she
why
is
he
having.
will do that.
No, she is co-author also, or you acknowledge?
Never.
Please go up.
At that time, she was my girlfriend, so
So
I cannot list her as a co-author.
No, no. I don't. No, that you can do, but we cannot do with our colleagues.
And at that time, we did not see each other. I was in the United States and working in Tom Jeffrey, Professor Tom Jeffrey's lab, because my boss does not have a structure perimeter. I have to borrow the equipment from Tom Jeffrey. He's a very kind man, very kind man. But I can only use this equipment at a very late night after his leaving, very late night, from 12 o'clock to the early morning.
Yeah.
And I can use the Skype, you know.
Yeah, Skype.
Yes, Skype. Video chatting with my wife.
Okay.
Yeah. She told me that how to calculate, how to use the Excel to do calculation.
Yeah, Excel, we have a expert with us. Rajiv Kapoor, Excel expert is there. But mathematics expert we don't have.
Still looking for.
But we will be looking for, like, you have given a lot of ideas. One thing also I just wanted to ask you that, like, we have almost completed this GIAN schedule, this tutorial. Okay. And I think many students, many participants, they have got some ideas. So how to proceed with that? Whether this type of interaction study, cellulose and cellulose, okay, they are done by the master's student, or you hire PhD student for doing these projects? How many PhD students?
I have only one, borrowed from our big boss,
Okay.
Professor Fu.
And you are
I am a co-supervisor,
okay.
not the single supervisor.
Okay.
So the project is determined by Professor Fu, not by me. Yes.
Okay.
I cannot determine. Okay, the topic, the PhD topic, I cannot.
And China bioenergy program, how much funds are normally given to R&D?
At least a million.
At least
A million.
million.
Yeah.
1 million.
Yeah.
1 million Chinese yuan.
More than that.
More than that.
Yes.
Oh, very good. Only bioenergy.
Yeah.
Okay. And the plants which you showed in,
But mostly hydrogen.
mostly hydrogen.
Hydrogen is highly supported. Yeah, highly supported. And the cell fuel, during the cell fuel.
Yes.
Yeah, in China, cell fuel is very popular with research. Yeah, cell fuel. Microbial cell fuel.
Lactis can also use as a
Yes. But microbial cell fuel depends on the production of hydrogen.
Here there are many applications where lactic also should be.
And if we can correlate the ethanol production with the waste management,
Yes.
the farming application will be much easier.
Yeah, actually the mandate of GIAN program is only to, not only delivering lectures, tutorial. This is also what we are doing.
Yeah.
Because we should know what China is doing to have a clean energy, and what efforts. Like I saw those big plants in the morning session. Can we have the slides of those plants? Yeah, please just show, because I was little busy in some administrative work that time, so I could not concentrate on that. It will be very nice if you can show it again. This is there, this one. Can use this. Show me plants. No, this can go.
This coming from this company they have an institute.
Okay.
Of their own. Every year they provide funding to the engineering university. This open the funding for education.
Okay.
For example, for the vehicle ethanol, vehicle bioethanol, hydrogen, They offer some class.
And this you are writing in Chinese.
Yeah, something in Chinese.
In Chinese. He's making notes in Chinese. So one thing, you told that this factory was started by your student.
This one, 2 students.
That is the name is Shina?
Jin Shan. This is the name of the company. You will next year you can have a opportunity to visit Guangzhou event. You can meet with them.
Okay, so what the company is doing well now?
Yes. Their technology is 100% on the low temperature treatment. It's a separation process.
Okay. So the only thing which is limiting factor for us is, especially in India, that we need to change the technology to the product.
Yes.
And sometimes we have the products.
After the treatment, they make a problem. They have a big problem because of the acetic acid. The residual acetic ions in the treatment solids in liquid, they cannot to remove it in acetic acid. So I developed a microbiome way to utilize all of the acetic acid. They consume the acetic acid and convert it into protein.
So normally,
It's a pestilence.
so normally in your this research program or master's program, you have one component of self-employment, right?
But the question, the scientific question originates from this. I will give them a topic, a lot of topic relates with technical problem, challenging.
And really, you can have a seat and so this one, like you have this. This
So
is
the
a
boss of this one is this company,
okay.
graduated from my school. That technology,
No, this is a company of a government or private company.
private, very large.
Very large.
They have stock in London. So stocks in London. The technology originates from China Academy of Science.
Chinese Academy.
They have an institute in Guangzhou,
Okay.
focused on energy, Guangzhou Energy Institute.
Raki is there. Raki, you can. I think we have already completed discussion. We'll take care of further suggestions and comments in another 15 minutes. Okay. So you just announce the finish option. If there is any questions to be answered or something, you just announce it. Please, thank you very much. And very productive session. Indeed, I think you have covered almost all the things today, including the technologies, including the various sectors. Yeah, and I think this was some session which has some technological relevance, and as well as the use of mathematical tools. You have showed us how the mathematical tools can be used. Yes, yes, that is very good. Very good. And I think we can close the session. Then we will proceed for tea. And tomorrow again we will meet in the morning at 10 start. 9:30 we should be there.
Break for tea and food.
I think because he has already finished, we are 15 minutes in advance. So that is not a problem. We can close it because so that we can discuss other things, and then we will proceed. So today's event is almost. So formal announcement by Raki.
So with this we come to the end of today's tutorial session. So that will be the end of the last tutorial session as well for the workshop. Now we'll have a tea break and we'll again reassemble here tomorrow at 9:30. Tomorrow we'll be having the closing ceremony.
Evaluation.